Imagine a population of butterflies where two forms of a gene exist. A, the dominant allele, and A, the recessive allele. How can we predict the genetic makeup of future generations in this population? That's where the Hardy-Weinberg principle comes in, a fundamental rule in population genetics. The Hardy-Weinberg principle states that, in a large, randomly mating population with no evolutionary forces at play, allele and genotype frequencies remain constant from generation to generation. It's like a genetic snapshot that lets scientists measure if evolution is happening, simply by checking whether the real world matches the principle's predictions. Let's see it in action with a concrete example. Suppose in a population, the frequency of allele A, P, is 0.7 and allele A, Q, is 0.3. Since there are only two alleles, their frequencies must add up to 1. P plus Q equals 1. To find the expected genotype frequencies, use the Hardy-Weinberg equations. The frequency of AA, the homozygous dominant genotype, is P squared. So 0.7 times 0.7 is 0.49, meaning about 49% of the population will be AA. For AA, the heterozygous genotype, calculate 2PQ. Multiply 2 by 0.7 by 0.3. That's 0.42, or 42% of population. Finally, for AA, the homozygous recessive genotype, it's Q squared. 0.3 times 0.3 equals 0.09, or 9%. If the real-world genetic makeup matches these predicted frequencies, the population is in Hardy-Weinberg equilibrium. If not, something, like selection or mutation, might be changing the gene pool. The Hardy-Weinberg principle gives us a powerful tool for tracking genetic changes and understanding the forces shaping populations. It's a cornerstone of evolutionary biology, helping us decode the story that genes tell across generations.